Good afternoon. So it's a real pleasure to have here today Dr. Iain Mattaj. And Dr. Iain Mattaj obtained the undergraduate degree in biochemistry at the University of Edinburgh, and then the PhD at the University of Leeds in UK. And he carried out the postdoctoral research in Switzerland, in the Bioscienze University Basel. And in 1985, he joined the AMBL in Heidelberg as group leader. And in 1990, he became coordinator of the gene expression unit at the AMBL, and later on scientific director, and in 2005 director general, and he was director general from 2005 and to 2018, and of the European Molecular Biology Laboratory, the European Laboratory world renowned for the outstanding research in molecular biology and for hosting a significant number of Nobel Prize winners, including the Nobel Prize in Chemistry for the cryo-EM. And the research that Dr. Iain Mattaj carried on gave relevant contribution in the field of ribonuclear protein particles that function in the processing of messenger RNA precursors. And the analysis of these ribonuclear proteins cover different topics, including the transcription of the RNA components, RNA protein interactions, ribonucleic protein function in messenger RNA production, and assembly of the ribonuclear protein from their components part in vivo. And then the work of Dr. Iain Mattaj led to numerous studies on the RNA and protein transport from the nucleus to the cytoplasm, and this group identified numerous factors and mechanisms involved in this important process. And more recently, Dr. Mattaj demonstrated the critical role of the GTPase run in the regulation of the mitotic events, including the assembly of spindle, the nuclear pore complex formation, and nuclear envelope assembly. Dr. Iain Mattaj obtained numerous prizes and honors, starting from, I have a very long list, starting from 1989, when he was elected EMBO member, and to this year, until this year, and when he was elected member of the Royal Swedish Academy of Sciences. So thank you very much for being here. It's a real pleasure and honor. Thank you.
Thank you very much, Francesca, and it's very nice to be here. In fact, I didn't realize until coming into the room, but I arrived in Milano in January of 2019, and to begin at the Human Technopole, and it was quite an adventure. There were 12 people, and essentially a few offices, and we were just beginning. And I was invited to come to BICOCA to talk at a systems biology conference, which had been organized. And I didn't know Milan at all. I had just arrived in Milano. I didn't, and BICOCA anyway is at the end of the universe. If you live, if you work at the expo site, it's a long way away. So I eventually found my way to BICOCA after many adventures on the metro, and got here late, of course, and gave a talk in this room. It was my first talk. It was a week after I arrived in Milano, I think, something like this. And I was feeling very disoriented. Anyway, I feel more comfortable a little bit now. And I'd like to tell you today what has been going on at Human Technopole since the beginning of January 2019. And what we hope to achieve in the relatively near future. So the mission of Human Technopole is very well defined, but it's actually very broad at the same time. It's to try to improve human health and well-being, including healthy aging, via contributions to fundamental research. So the idea is to be an organization, an institution that carries out fundamental research, but also creates research infrastructures that include infrastructures that will enable approaches and potentially new approaches to personalized or stratified medicine, and to try to do this in a collaborative way with the scientific community, both nationally and also, of course, internationally, and to work together, and I'll come back to this probably several times during the talk, both with academic partners, but also with medical partners and industrial partners to try to create a hub. And the ambition of the Technopole plan and project was large. It was to be able to have around 1,000 scientists, which means in total this would be an institute of maybe 1,400 scientists on a single site. And just, you work in a university, so these numbers are tiny for you. But in terms of a research institute, the biggest research institute in Europe at present is EMBL. It has 1,800 people. The Crick Institute has roughly 1,200 people. So we're talking about an ambitious plan and a large-scale plan. We obviously have a set of values by which we hope to act. What we want to do is to make sure that everything we do meets the criterion of scientific excellence, that we are reviewed and we make use of evaluations to ensure the quality of what we do. We will be an institute in the life sciences broadly, but we want to be within that scope interdisciplinary. We want to be diverse, we want to be international, and as I said, we want to be collaborative. And we also have a plan to provide, through building infrastructures, service to the national and international community. So I won't go through this in great detail, but there are many aspects in which a research institution can be open and can be helpful to the surrounding community. I've talked about infrastructures and facilities. Visitor programs, enabling people to come to either to collaborate or to make use of the infrastructures. Training programs, we have begun to carry out training programs, obviously virtual training programs in advanced technologies, and this is something that we will want to expand on. Fostering international collaboration, some of the people that we have hired already have or are now submitting projects, networking projects to the European Commission. And we want to involve other national and international labs in these networks to try to foster the inclusion of more people in international grant funding. And I've already mentioned that we will be engaging in technology transfer, and we hope to continue to engage with companies, and I'll come back to that because the opportunity is really there. So the I was not involved in making the original master plan, as it was called, for Human Technopole. This was a large-scale effort which was led by the IIT in Geneva, who worked together with many people from the community to come up with a plan that, would have taken billions of dollars and lots of people to implement. So the plan was big, it was very broad. And the first task for us in implementing something was to select from the many different parts of the proposal the things that we should do first and that we should begin with. And I think, and this is not rocket science, I think that we have to take the opportunities which arise at particular points in time. And in my opinion, there are two key technologies which have really changed life sciences in the last 20 years. One over the last 20 years, one in the last 8 years or so. And the first of these technologies, of course, genomics. So having the possibility to sequence the genomes of human beings makes us into fantastic experimental animals, because previously we were very limited in how we could analyze the human system. And now there are more and more both experimental but also computational technologies for studying human beings. So for me, genomics was absolutely a core concept that we had to embed into Human Technopole. And specifically, genomics has been super successful in some areas. Think of cancer research. That we know so much more about cancer as a family of diseases than we did prior to the genomics era. But other aspects of life science have been more difficult to study in this way, and there are very good reasons why they've been more difficult. And I think neuroscience is one of these areas. There are areas of neuroscience which are very advanced and which are very, very exciting. But actually, the understanding underlying mechanisms through use of genomics and biochemistry in neuroscience has not really been easy. And the reason's obvious. You can't cut off somebody's brain and study it in the same way that you can cut off their tumor and study it. So there are good reasons for this, but I think there are also reasons why this is changing. And so neurogenomics was a particular area. Structural biology was, in my mind, the reason for choosing this was partly based on what you can do using the tools, but partly also based on the development of cryo-electron microscopy, which has revolutionized structural biology over the last 8 years or so. And those sciences, like many other areas of the life science, produce huge amounts of data. And so we need computational scientists to develop tools in order to be able to work with these kinds of data. And the final idea, again, this was part of the concept of Human Technopole before I had anything to do with it. But this was an idea which I thought was a really good one, which was to take these methods of big data analysis and to apply them not to individual disease states, but actually to apply them to the health system and the healthcare system itself. And that, I think, is a very good idea. And this part of Technopole actually had begun before Technopole existed, because there was a project between IIT and the Politecnico di Milano, which was the beginning of this area of research. And we are continuing this. IIT, of course, has stepped back from Human Technopole, but we're continuing this with the Politecnico. So, and who is going to do this? Does anybody know how I can can get rid of espandi il pannello per visualizzare il video. Non ho un video. Anyway, so basically as I said, there were 12 people in total when I arrived. And so the first thing we had to do was to begin to hire people. Hiring has gone very well, both in administrative areas, science support areas, and in the science. It's fine like that. Thank you. And these are the people we've hired thus far. Actually, this goes out of date very quickly, and it's not quite up to date. But these are the people we've hired so far, who are either the more senior researchers. So these are the people who will head or co-head the research centers at Human Technopole. And these are group leaders who have joined one or other of the research centers. And in just a little bit more detail, in genomics, we have Piero Carninci and Nicole Soranzo. Nicole is a population geneticist. Piero, I would guess you all know, has developed technologies that enable functional genomics to be done. Giuseppe Testa is the exception that proves the rule. He's the only person who we actually recruited from inside Italy. He was working at Yale, and he moved over to become the head of neurogenomics. Andrea Satoriva is the head of computational biology. Alessandro Fanini and Gaia Pigino, structural biology, and our most recent senior recruit is Emanuela D'Angelo Antonio, who is the head of health data science. And those people all, except Alessandro, who's actually, even though you would never guess, he's actually British by birth. All the rest are Italian by nationality, but have been outside of Italy for a very long time. Piero is the record holder. He's been outside of Italy for 26 years and is coming back. The group leaders are more diverse. So Nereo is from Croatia, Jose is from Mexico, Elena is Italian, Oliver is Dutch, Lorenzo is Italian, and so on. But the group is quite diverse, both in terms of expertise and in terms of where they come from. And we hope to be able to continue to recruit people of this caliber from all over the world. In terms of facilities, we're building genomics facilities, a cryo-EM facility, light imaging, a facility to automatically reprogram iPS cells and create organoids, and on the computational side, an image analysis facility and a data center. I want to give a little view into some of the research that the people who are coming or are here are actually doing. So this is a paper which is from Nicole's group, and it is about the effect of mitochondrial DNA variants on the concentration of N-formyl methionine and its link to late onset human disease. So the human mitochondrial DNA encodes 13 electron transport chain proteins, as well as quite a lot of the machinery for their transcription and translation in the mitochondria. You probably are aware that many human diseases, or a large-ish number of human diseases, are caused by mutations in the mitochondrial DNA rather than in the genomic DNA. And particularly late onset diseases are associated with mitochondrial DNA variations. And so Nicole's group participated in the interval study, which is a cohort study looking at 16,000 healthy people and looking at over a long period, they've been studied for 20 years or so, this cohort of people, looking at blood-derived markers and correlating them with mitochondrial DNA variation. And the conclusion, or one of the conclusions that they came to, is that relatively common mitochondrial DNA variants are associated with increased levels of FMET in the blood. And what these are geneticists. The data is always incredibly derivative and complicated to explain. But basically, this dotted line here is the significance line of the association of a particular variant with an increased level of a specific. Anyway, all of these black dots are specific metabolites. And the ones that are marked in red have got a significant variation in the FMET levels correlated with a specific mitochondrial DNA variant. The red ones are FMET, and what you can see is that a lot of the really significant correlations and changes are with FMET levels. And this was a surprise. This came from an analysis of 20 years worth of patient samples. So what they went on to show was that FMET levels in general, but very sensitively and very extremely in correlation with these mitochondrial DNA mutations, affected protein synthesis and transcription. So transcription was increased generally in the context of these variants. That was not affected by adding exogenously, by increasing the concentration of FMET. That's what's shown on the right of this slide. And on the left, what you see is the level of translation, and it's in relation to FMET, exogenous FMET. The cytosolic, that was mitochondrial translation. Okay, anyway, not just mitochondrial protein synthesis, but also cellular protein synthesis is very sensitive to FMET compared to controls in these mitochondrial DNA variant cells. And this is probably related to stress responses. Anyway, so let me just cut to the chase of this particular study, which is that those variants, those mitochondrial DNA variants that cause elevated FMET, which is what's measured by these boxes, are very frequently predictive of late onset disease. And the data to support this comes from the study of this cohort, which has been under study for 20 years. So the data comes from the FMET levels of the people who started normally and developed late onset disease. And you can see kidney disease, coronary disease, heart disease, other types of heart disease, stroke, arterial lung disease, and death are all correlated with increased FMET levels. So essentially, doing this profiling put the group onto the track of an unexpected effect of mitochondrial DNA mutations and helped them towards the beginning of an understanding, a molecular understanding of the basis of the effects of this disease and the increase in FMET concentration. So the others are much simpler and they're pictorial, so hopefully we will go better. So The synthesis of ribosomes is a very complicated process. I'm sure you all know it happens in the nucleolus. It's a mega multi-step process where you create large pre-ribosomal subunits, which then actually get smaller as they mature towards ribosomal subunits and eventually are transported to the cytoplasm. And this is a paper by Philip Erdmann, who's one of the group leaders we've recruited, which he carried out the work while he was in Wolfgang Baumeister's lab in Munich. And essentially, ribosome biogenesis has previously been studied through the study by electron microscopy and other techniques of isolated precursors. So quite a lot is known. People like Ed Hurt and David Tollerby worked for many, many years on aspects of ribosomal RNA biogenesis, ribosomal subunit biogenesis. So what Oliver wanted to do was to look in the native environment at this process. And so he used for this Chlamydomonas cells because they're quite flat and therefore quite suitable for doing cryo-electron tomography, and looked to see where he could see pre-ribosomal subunits. And they were clustered on the surface of the nucleolus. So you can identify these by their shape by comparison to the previous studies that were done with isolated particles. And this gives you some idea of what the big stuff looks like in a slice of a Chlamydomonas cell. So it's very dense, it's very full of particles. But by doing electron tomography, you can actually identify and isolate the particles. And what Oliver was able to show is that pre-ribosomal particles that are identifiable in the nucleus get larger as they leave the nucleolus and as they cross the nucleoplasm and cross the nuclear envelope. So there are two steps at which they get larger. So looking in more detail at the individual particles, you can look at families and get an average structure. You could see that both for a small subunit and large subunit, you could put them into classes that were more mature and more close to the mature ribosomal subunits. And basically, this seems very logical. It's a lot of work to come to a logical conclusion. They get more mature as they cross the nucleoplasm and come to the nuclear envelope. So that was this study. And the final study is one by Gaia Pegino and her collaborator Karsten Janke from the Institut Curie in Paris. And it's about the effect of tubulin glycylation on male fertility and the flagellum. So it has been known for many years that tubulin has many different post-translational modifications. And there are only very few for which a clear function has been obtained. And Karsten Janke has been very interested for many years in tubulin glycylation, which till now had no defined function, even though it's clear that it has a phenotypic effect. And I'll just go through this. So what Karsten found first, looking in mice, was that loss of tubulin glycylation affects fertility. So there's a big effect on male fertility, not female fertility. And this is correlated with a loss in sperm motility. And when they looked in detail, and this was part of Gaia's work here, when they looked in detail at sperm swimming, so normally sperm are very intent on their goal. So they move straight towards the egg. They're attracted, obviously, by chemical signals, and they follow the path of concentration of the signal. But in glycylation-related mutants, the behavior becomes either spiral or circular. So this is why fertility is reduced, because the sperm don't know where they're going. And so they don't reach the egg in most cases. And Gaia went on to study this at high resolution. And this is a reconstruction of the motors that are associated with the sperm axoneme, which has microtubules along its center, and showing that the motor protein dynein and different subunits of dynein is not associated in a normal way with these sperm flagella, and therefore this is the mechanism of the lack of directionality and therefore reduction in fertility. And I won't go into more about this. I've forgotten how long I was supposed to speak for. Okay, so I want to say a little bit more about the type of thing we would like to do with the community. These projects, two were quite large-scale collaborations. The cohort study and the tubulin study all involved several laboratories, and obviously the people who generated the cohort. And we'd like to continue to work in this way by either helping put together or joining consortia of people who have a particular larger scale goal in mind. And what we've done so far is to join a couple of existing cohort studies, one of which is about 27 years old now. It's the Molisani cohort from Molise. It's a cohort study, so this is just a group of 25,000 healthy individuals who have been followed throughout their life. Their health records have been kept. They go every couple of years to have blood samples and urine samples, etc., taken and to have a medical examination. And the data is collected. And again, the idea is that eventually you can connect either environmental exposure, behavior, or genetics to disease which comes later, and perhaps use the data that you've collected to have early onset predictors. So this is one study that we will join because it has been impossible to have either genetic or genomic data from these patients so far. And it's a very well organized study. There's lots of data collected, and we would like to add the genetic and genomic data, and the organizers are very happy to work with us in order for us to complement their work by providing this. Similarly, with the Oasi Maria Santissima in Sicily, which is an IRCS, they have a cohort study, but this is disease specific. It's a cohort of people who suffer from autism spectrum disorders. And here again, this is a well-organized study. It's not nearly so big as 1,500 people in it. But there again, there is no genetic or genomic data. And so we want to work with them to add this to the data set which they've collected. And then just because of what happened since we started Human Technopole, we've obviously become involved in various COVID-related studies. Here, our role has been mainly as a provider of genomic information to look at some groups of patients for whom it was interesting to have genomic information, or to contribute single-cell transcriptomic data to studies of the immune system. which are being done. And this is being done with a whole set of different partners from Padova, from Rome, from Milan, etc. And I won't go further into that. We will carry out research, and so we'll obviously need to have internal training programs for undergraduates, PhD students, and postdocs. We also are hiring people straight from postdoc, and so we also have to provide training and mentoring to the young PIs as they learn how to run a research group. In addition to this, we want to organize external training events, courses, conferences, and workshops, and as I said, have scientific visitors coming to Technopole. So we have joined a couple of existing PhD programs. Obviously, we cannot give, we're not a university, we can't give PhD degrees. And so we have joined other partners, including universities, to become part of existing PhD programs, which who invited us basically to join. One of those is the SIM program, which is based in Milano and Naples. The other is a program for essentially computational analytical methods, which is based at the Politecnico. In terms of postdocs, we have, again, a relatively small cohort to date of postdoctoral fellows. 10 of them come from Italy, 6 come from other countries, and obviously we're in the process of building up this group of people in particular. And despite the problems of COVID, Florian Hug has led the development of 2 training courses so far on advanced image analysis methods. So these are computational methods, and therefore organizing those virtually was relatively straightforward. And he organized one in 2021, and there will be another one actually, they both say 2021, but the next one will be in 2022. I think it's very important that organizations which are carrying out research also have to be aware of the link between research and innovation, and to try to promote this link. It's a very important step. It's also, quite honestly, it's the reason why we get funded to do research is because the people who give us the money, the taxpayers or the politicians who decide the money should be spent this way, want through research to have the basis for carrying out innovation. And I think it's therefore essential that research organizations engage in innovation in an active way. So the first thing that we will organize here is a course in entrepreneurship for young PIs, for young principal investigators. And this, unfortunately, has been a COVID victim because this has to be done in person. And it has been very, very complicated. We've had to keep delaying this initiative. But at the moment, it looks as if we'll be able to do this early next year. And finally, I come to the Early Career Fellowship program and to a short explanation of why we're doing this. So the idea for the Early Career Fellowship program, again, like many things, it wasn't an original idea that came to us. There are programs called the Harvard Armenise Fellowships and the Rita Levi Montalcino Fellowships of Moore, which previously tried to do exactly the same thing. And I met some of the people who had been evaluators for the Harvard Armenise program. And what they said is that this is a program just like the one I'm going to describe. We copied this with their help in order that we didn't have to reinvent something that already existed and was successful. So what they said is that this is a fantastic program. People really want to be part of this program, but there are very few fellowships. I think the Rita Levi-Montalcino Fellowship was one every few years, basically, and the Harvard Armanisa was one or two per year, and not every year. So there seemed to be a group of people who wanted to come to Italy to set up an independent laboratory and who were waiting for a program which would fund them to make this easier. And so we decided to try to set this up. We talked to Moore about doing this, and they were very supportive, and morally supportive, not financially supportive of doing this, carrying out this program. And so we developed it together with them, and as I said, with help from the people at Harvard Armanisa. And the idea is to provide some funding to very promising young scientists to set up an independent lab. And the concept is obvious, that people who come from their postdoc in the life sciences are generally at the most productive, most creative stage of their career. And the idea is to provide more of them with the opportunity to set up something that's really independent at that stage, rather than to join someone else's lab and essentially lose their most creative period. So Ilaria, who is in the front row, organized this with her colleagues in the training program. And we have now selected the first 5 fellows with the help of mainly external scientists from other countries, but also with the help of scientists from this country, one of whom was from the Statale and was appointed by Moore as a member of the evaluation committee, and 2 of whom are members of our management committee, Stefano Piccolo and Irene Bozzoni, for those of you who know them. So these are the 5 fellows: Veronica, Mirka, Carmen, Gabriele, and Daphne. And they came through the selection process and impressed us all, and have now selected and made agreements with host institutions. So obviously Veronica, who is sitting in the audience, is coming here. And we're very, very happy that that is the case. Gabriele is going to Humanitas from the ETH in Zurich. Daphne is still in New York, but will come at the beginning of the year to Geneva. Carmen will come to Trieste from UC Davis at the beginning of the year. And Mirko is in the process of moving definitively from Heidelberg to Naples to join TGEM. So they will begin from January 2022. We are quite happy with the geographical spread. We're very happy that both research institutions and universities will be host institutions for the fellows. The idea that we had and the things that we thought we could contribute, apart from the selection and financing, were that these people were selected to work in the areas in which Human Technopole will be active. And so we are hoping that they will essentially become associate faculty and will come to retreats, will work together, will collaborate, will make use of the facilities that we will have. But of course, the main goals are to help relocate more talented young researchers to Italy. And we think that those people will also serve as bridges between us and other institutions, because as I've emphasized throughout, we want to work together with people, and we think having living bridges between us and other institutions will help us to make good links. So that was it. Thank you very much.